Bismillahirrahmanirrahim. Assalamu alaikum to all of you. I am again back with a new lecture, that's the embryology of the human air. So we'll be discussing the developmental events, how the air is being structured in an embryo with the actual time periods and the congenital anomalies, right? So when we look at the adult air, we find three distinct regions: the external air, middle air, and the internal air. Right? The external air does the function of sound collection. Right? It collects the sound waves and convey it to the inner portion of the air. The middle air, it is a sound conductor from external to the more deep tissues of the air, that is from the external air to the internal air, right? And the internal air, it converts those collected sound waves into the nerve impulses and then register their changes in the equilibrium or balance, right? So the main function is actually done by the internal air, right? So this is how the human ear looks like, having the external ear. This region is the external ear, this is the middle ear, and this is the internal ear, right? So let's discuss about the internal ear first, that how it is produced. When we look at the embryo, at approximately 22 days old embryo, we can see the thickening of surface ectoderm near the rhombencephalon. Here we can see this. This is known as otic vesicle. Right, जिस तरह eyes का optic vesicle होता है, likewise for ears we have the otic vesicle, which are the thickenings of surface ectoderms near rhombencephalon at approximately which day, 22nd day, right? इनको हम otic placodes भी कहते हैं. Now what happen, these otic placodes, they will invaginate rapidly, forming the auditory vesicles or otices, right? In this picture you can see these are the placodes. अब ये जो इसका blue tissue दिख रहा है, it will invaginate more, ठीक है, and then it will form the otic vesicle, right? During this development, now this vesicle will divide into the anterior and posterior component. जो anterior component है, which is the ventral component, it will give rise to saccule and cochlear duct. The posterior component or dorsal component will give rise to the utricle, semicircular canals, and endolymphatic duct, right? Together, these epithelial structures form the membranous labyrinth. इफ़ यू कैन रिकॉल, आपने ज़रूर इंटरनल एयर में पढ़ा होगा मेम्ब्रेनस लेबिरिंथ, बोनी लेबिरिंथ एंड ऑल, राइट? सो द एंटीरियर एंड पोस्टीरियर कंपोनेंट्स, इन ये कहाँ पे डिराइव करके कौन कौन से स्ट्रक्चर्स बना रहे हैं, दिस इज इम्पॉर्टेंट. सो एंटीरियर कंपोनेंट विल फॉर्म द saccule and cochlear duct, this is the anterior component, and the posterior will form the utricle, semicircular canals, and endolymphatic duct. I'll show you in the picture. Here we can see this is the saccular portion, this is the utricular portion, right? And here we can see that how the saccular portion is giving the outgrowth, the tubular outgrowth, which is turning into the circular canals known as cochlear duct, right? तो इस तरह से cochlear duct saccule से आगे आगे बढ़ती है, right? So in the 6th week of development, the saccule forms a tubular outpocketing at its lower pole, which we have already discussed, like this. ठीक है, ये सेक्युलर पोर्शन उसी से आया, ओटिक वेसिकल से, ठीक है. अब उसी से एक ट्यूबुलर आउटग्रोथ आई. नाउ दिस आउटग्रोथ इज द कॉक्लियर डक्ट, विच विल पेनिट्रेट द सराउंडिंग मेसेनकाइम इन द स्पाइरल फैशन, ठीक है. और ये कब तक होता रहेगा, 6 वीक से ये ट्यूबुलर ग्रोथ आई है. बाय द एंड ऑफ 8th वीक हमारे पास प्रॉपर कॉक्लियर डक्ट विल बी फॉर्म्ड. When the proper 2.5 turns are completed of the cochlear duct, its connection with the remaining portion of saccule is confined to a narrow pathway. इसको हम नाम देते हैं कि ductus reunions. ठीक है, जो ये ट्यूबुलर ग्रोथ थी, जिस तरह से कॉक्लियर डक्ट बनने लगी, जब 2.5 टर्न्स हो गए, नाउ इट इज़ elongated that much that अब इस सिक्यूल के साथ इट इज़ जस्ट कनेक्टेड थ्रू अ नैरो connection which is known as ductus reunions, right? Now the surrounding mesenchyme जो इसके आसपास था, अब वो जो है differentiate हो जाएगा into the cartilaginous tissue, right? Now in the 10th week, this cartilaginous shell, it undergoes vacuolization and two perilymphatic spaces are formed. Now look at picture A first. This is the cochlear duct, right? This is the surrounding mesenchyme. If you can see the dark gray color, ठीक है? अब इस surrounding mesenchyme में दो spaces, दो vacuoles आ जाएंगे, right? ये दो vacuoles जो हैं हमारे पास ये एक हो जाएगा स्केला वेस्टिबुलाई and this will be the scala tympani, right? So scala vestibuli and scala tympani के बीच में अब हमारे पास cochlear duct हो जाएगी. जो cochlear duct है, it is separated from scala vestibuli by vestibular membrane. यहाँ बीच में vestibular membrane होगा. C में देखो, this is the vestibular membrane, right? And the cochlear duct is separated from scala tympani by the basilar membrane. This is the basilar membrane. सही है. So the basilar membrane separates cochlear duct from the scala tympani. The vestibular membrane separates the cochlear duct from scala vestibuli, right? And what are scala vestibuli and tympani? These are the two vacuolated spaces produced in the cartilaginous shell around the cochlear duct, right? The lateral wall of the cochlear duct, it is found attached to the surrounding cartilage by spiral ligament. ठीक है, इसका जो lateral end है, this is the lateral edge, right? ये जो है इसके surrounding cartilage के साथ, अब हम इसे surrounding mesenchyme नहीं कहेंगे, it is the cartilaginous shell, right? इसके साथ it is attached via spiral ligament. This is the spiral ligament, right? Whereas the median portion, it is connected to the cartilaginous rim by a long process, क्योंकि ये ऑलरेडी इस साइड पे आ चुका था, राइट? तो ये एक लॉन्ग प्रोसेस के थ्रू कनेक्टेड होगा टू द cartilaginous rim. This connection is known as modulus, right? This modulus is actually the future axis of the bony cochlea. ठीक है. Now the epithelial cells of the cochlear duct are alike, एक जैसे होते हैं, ठीक है? बट विथ फर्दर डेवलपमेंट उसमें दो रिजेस बन जाएंगी. यू कैन ईजिली सी ओवर हियर, यहाँ पे कोई सिग्निफाइड रिजेस नहीं है. यहाँ पे अब ये दो रिजेस में अपने आप को कंपाइल कर लेंगे, द आउटर रिज एंड द इनर रिज, राइट? द इनर रिज विल फॉर्म here, yeah, the inner ridge will form the future spiral limbus. ये spiral limbus बनाएगा, and this is the outer ridge. Outer ridge will actually form the hair cells, rows of hair cells बनाएगा. These are the sensory cells of the auditory system. So the epithelium of the cochlear duct will form two ridges, the outer ridge and the inner ridge. Outer ridge will form the hair cells, the rows of hair cells, and the inner ridge will form the spiral limbus, right? What's the significance of this? We'll talk about it in the upcoming slides. Now we can see that this epithelium is covered by a fibrous by a fibrillar gelatinous substance known as tectorial membrane. ये एक membranous gelatinous membranous material है जो epithelium के ऊपर है. This is tectorial membrane. This tectorial membrane, now see at this this B part of the picture, we see that this tectorial membrane is attached to the spiral limbus, right, and that rests with its tips on the hair cells. ठीक है, अब क्या है स्पाइरल लिम्बस? स्पाइरल लिम्बस वॉज द इनर रिच, और ये हेयर सेल्स किससे बने थे? आउटर रिच पे। तो जो आउटर रिच से हेयर सेल्स बन गए और जो इनर रिच का स्पाइरल लिम्बस है, स्पाइरल लिम्बस से अटैच होगी टेक्टोरियल मेम्ब्रेन दैट विल बी एक्टिंग लाइक एन अम्ब्रेला ओवर द हेयर सेल्स। ठीक है, दिस इज द umbrella of tectorial membrane, right? Now these sensory hair cells and the tectorial membrane, this all together is the organ of Corti, right? So what is organ of Corti? Organ of Corti is actually the combination of sensory hair cells formed by outer ridge of cochlear duct epithelium and the spiral limbus, which is formed by the inner ridge of the epithelium, which is attaching the tectorial membrane to its the one part of tectorial membrane to itself, right? So this tectorial membrane and the sensory hair cells are forming the organ of Corti, right? Now we'll talk about the utricle and semicircular canals. During the sixth week of development, the semicircular canals will appear as flattened out pocketings. ठीक है, जो utricle part था उसका ओटिक वेसिकल का, राइट? इस तरह से 3 आउट पॉकेटिंग्स आएंगी। इनके जो आउट पॉकेटिंग्स हैं उसमें सेंट्रल पोर्शंस जो है इसका diminish होने लगेगा, and the time will come when this whole central portion is diminished and proper 3 semicircular canals are being formed. When we look at the canals, each end of the canal has a dilated end known as crus ampullaire. ठीक है, और जो remaining पतला वाला part होगा that will be crus non ampullaire, right? Now the cells in the ampullae form a crest, the crista ampullaris. सही है, जो इसके अंदर होंगे एम्पुली में वो क्रिस्टा एम्पुलरिस होंगे, जिसके अंदर सेंसरी सेल्स होंगे फॉर द मेंटेनेंस ऑफ इक्विलिब्रियम, right? Similar in sensory areas, the maculae acoustici, they develop in the walls of utricle and saccule, right? Then we come to the tympanic cavity, which originates in the endoderm, and it is derived from the first pharyngeal pouch, right? This pouch expands in the lateral direction, and it comes in contact with the floor of the first pharyngeal cleft. Let's see in the picture over here, like this. Okay. The distal part of the pouch, the tubotympanic recess, this is the distal part of the pouch. It widens and gives rise to primitive tympanic cavity. Okay. And proximal part remains narrow, forming the eustachian tube. This picture is very nice. Here we can see this is the tubotympanic recess. इसका distal part अब dilate हो जाएगा, forming the tympanic cavity for the middle ear, and this proximal part will be the narrow part which will form the auditory tube, right? So this is how the tympanic cavity has been expanded, and this is the proximal narrow part forming the eustachian tube. Now, the ossicles within the tympanic cavity that are malleus, incus, and stapes, these are derived from the cartilaginous tissues of the first pharyngeal arch. ठीक है, malleus, incus first pharyngeal से होंगे, stapes will be from the second pharyngeal, and these are formed during the embryonic period, but they remain embedded within the mesenchyme till 8th week. Means इसके surrounding mesenchyme रहेगा, ठीक है. Now with time the mesenchyme will dissolve, providing the space for primitive tympanic cavity to expand. ठीक है। इसके आसपास जो मेसेंकाइम है अब वो डिसॉल्व हो जाएगा ताकि टिम्पैनिक कैविटी और प्रॉपरली एक्सपैंड हो सके। व्हेन द ऑसिकल्स आर एंटायरली फ्री फ्रॉम द मेसेंकाइम, द एंडोडर्म epithelium connects them in a mesentery like fashion. अब ये sort of ligaments के साथ attach हो जाएंगे क्योंकि mesenchyme चला गया है, now they are free in the environment in the cavity, so there must be something that will hold them into the places, so that's the supporting ligaments, right, within the mesenteries. ठीक है, showing the tubotympanic recess जिसका proximal part narrow हो जाएगा और distal part dilate हो जाएगा, right. See, proximal part narrow, distal part expanding, forming the tympanic cavity, and then these cartilaginous models, the ossicles will come inside the cavity. ये हमारे पास surrounding mesenchyme है, ठीक है. And in this picture we can see that the surrounding mesenchyme has been vanished, expansion of tympanic cavity has taken place, right. Now we'll talk about the muscles and the mastoid ear cells. Because the malleus is derived from first pharyngeal arch, its muscle, the tensor tympani, it will be supplied by mandibular branch of trigeminal, and stapedius muscle, which is attached to stapes, it is innervated by facial nerve, which is the nerve of second pharyngeal arch. Remember, we have done these muscles in the gross section as well. Now, during the late fetal life, the tympanic cavity expands dorsally, means posteriorly, by vacuolizing the surrounding tissue to form the tympanic antrum. And after birth, the epithelium of the tympanic cavity invades the bones of the developing mastoid, and epithelium lined ear sacs are formed, right. So this tympanic cavity, the epithelium of this will invade the mastoid processes and form the ear cells like pneumatic bone बन जाएगी, right. Then the external auditory meatus, it develops from the dorsal portion of the first pharyngeal cleft. We can see over here, this is the first pharyngeal, and it is going dorsally deep inside, ठीक है. तो ये हमारा बना रहा है external auditory meatus. At the beginning of the third month, epithelial cells at the bottom of the meatus proliferates, forming a solid epithelial plug known as meatal plug, ठीक है. ये meatal plug रहता है. In the seventh month, this plug dissolves and the epithelial lining of the floor of the meatus participates in formation of the definitive eardrum, right. तो सेवन्थ मंथ में ये प्लग जाना शुरू कर देगा और यहाँ पे हमारी टिम्पैनिक मेम्ब्रेन बन जाएगी. The meatal plug, if it persists, so it will result in the congenital deafness, because there will be no drum-like structure that will vibrate that will conserve the sound waves, and it will be transferred further, right. So when we talk about the eardrum or tympanic membrane, we have already discussed कि वो बाहर की साइड में उसके पास ऑडिटरी ट्यूब की एपिथेलियम होगी और इनर साइड पे उसके पास tympanic cavity की epithelium होगी, right. So outer is the ectodermal epithelial lining and inner one is the endodermal epithelium lining, right. And in between these two we have already discussed connective tissue fibers होंगे, connective tissue होते हैं, right. So the major part of the eardrum, if we see, is attached to the handle of malleus. ये handle of malleus होता है, ठीक है. और बाकी जो होगा, it is actually forming the separation part which is separating the outer ear from the middle ear, right. Now we'll discuss that how the auricle is developed. The auricle develops from 6 mesenchymal proliferations at the dorsal end of first and second pharyngeal arches. Here we can see these are the 6 mesenchymal proliferations, right, around the first pharyngeal cleft, first and second arches के around it, right. इन स्वेलिंग्स को हम ऑरिकुलर हिलॉक्स भी कहेंगे, ठीक है. एंड इन द लेटर लाइफ these hillocks which are three on each side of the meatus, they will transform in such a shape that they will form a proper external pinna, external ear, right. इनिशियली एक्सटर्नल एयर्स ये हमारे लोअर नेक रीजन में होते हैं, राइट. एंड एज द हेड ग्रोस एंड एज द मैंडिबल ग्रोस, दीज दे राइज अप टू द लेवल ऑफ आई, ठीक है. ये हमारे पास नॉर्मल पोजिशन ऑफ एयर्स आ जाती है. Now we'll talk about a little of the abnormalities. When the congenital deafness occurs, it is usually associated with deaf mutism, which is caused by abnormal development of the membranous and bony labyrinths, or by malformations of the auditory ossicles or eardrum, right. In most extreme cases, the tympanic cavity and external meatus are absent. Most forms of the congenital deafness are caused by genetic factors, but there are few in which environmental factors are also reported, right. So these environmental factors, they might interfere with the development of internal or middle layer, right. It is known that the rubella virus, if this virus affects the embryo in the 7th or 8th week of development, it may cause damage to the organ of Corti. Okay, now my question is, what is organ of Corti? How many of you have answered that it's the combination of tectorial membrane and the sensory hair cells? जिसने आंसर यही दिया है, दिस इज़ करेक्ट. और जो वापस से स्लाइड्स पे चला गया है, तो इट मीन्स आप लेक्चर सही से नहीं सुन रहे थे. सो यू हैव टू गो थ्रू इट अगैन एंड अगैन, राइट. नाउ लुक एट दिस पिक्चर्स. इसमें हमें क्या दिख रहा है? द एरोज आर शोइंग सम पिट्स, राइट, डिप्रेशंस हैं. और नीचे सी और डी में वी कैन सी सम प्रोट्रूबरेंसेस, राइट, near the ear or pinna. Okay, so these are the defects, external ear defects, which are common. They include minor to severe abnormalities. जो अभी हम प्रोट्रूबरेंसेस की बात कर रहे हैं, those are actually known as pre-auricular appendages, right? और जो डिप्रेशंस थी उनको हम कहते हैं pre-auricular pits, right? So these are the skin tags or shallow depressions. Pits are the depressions. Appendages are the skin tags that are lying on the anterior region of ear. तभी इसका नाम pre-auricular है, right? Pits may indicate abnormal development of the auricular hillocks, right? और appendages भी ये है कि accessory hillock बन गया, ठीक है. 6 नॉर्मल है, एक मोर देन 6 है तो accessory hillock बन गया. और इसकी abnormal development, अगर abnormal resorption होके अगर देखा जाए तो pits बीच में रह सकती है, ठीक है. And these external ear defects, they might be associated with other malformations as well, right. So this was all related to the embryology of the development of hair. For you people, the important things to memorize in this is the main time periods, कि किस मंथ में कौन से एयर का कौन सा स्ट्रक्चर बन रहा है, राइट. मिडल एयर कब बन रहा है, इनर एयर कब और कैसे बन रहा है, एंड एक्सटर्नल एयर कब एंड कैसे बन रहा है. ऑर्किला हिलॉक्स हैं तो वो कौन सा पार्ट ऑफ एयर बना रहे हैं. कॉक्लियर डक्ट है, सेमीसर्कुलर कैनाल्स हैं, वो किस पार्ट से बन रहे हैं. व्हाट इज़ ऑर्गन ऑफ कोटाई, राइट. इक्विलिब्रियम और बैलेंस कौन सा पार्ट ऑफ एयर करता है, right. So these questions are important from this part of lecture. You don't need to worry about this, ठीक है. Slides, you might find the slides heavy, as my lectures, my slides in my lectures are not so heavy. I have put up this matter because embryology का lecture या तो मैं सामने पढ़ाऊँ, explain करूँ बार बार, that way is easy, or I can do this way, that I put much of the text in the slides so that you can read it properly along with listening to my lecture, right? So that is why these slides are a little heavy. So don't take these slides as boring slides. Keep reading it again and again. Keep revising it again and again so it becomes easy for you people. If you want to do it from books, that's fine. Otherwise, this presentation is completing all the important aspects of this development of air thing. So I guess that's all for today, and I hope everything is clear to you people. Important things I have discussed with you कि इसमें से क्या क्या आपसे पूछ सकते हैं, right? So I hope that you are okay with this, but still if you have any query, you can email that to me, right? And I'll inshallah reply that to you. Okay, till then, Allah Hafiz.